Very warm welcome to today's webinar to all of you. Very good morning. Thank you for joining us today. I am Himashri and I am Senior Executive at DPT Wellcome Trust India Alliance, and I will be your host for today's workshop. I'm glad I can see that a lot of attendees have already joined, so we'll now start. So today we bring to you the fourth lecture of the Continued Biotechnology Education Lecture Series. These lectures are organized by India Alliance in collaboration with Guru Angad Dev Veterinary and Animal Sciences University, in shortly known as GADVASU, and Indian Institute of Technology Roorkee, that is IIT Roorkee. Our aim is to bring to you a diverse range of topics in biotechnology. The first three lectures of the series focused on genetically engineered vaccines, next generation sequencing, and metagenomics. Today, our topic is on genome editing in animals for enhancing productivity. So, next slide, please. Let us start with a quick introduction to the organization that I represent, the DBT Wellcome Trust India Alliance. India Alliance is a charitable trust registered in India that funds research in health and biomedical sciences. So yes, just like the DST, the DBT, the CSIR, we do fund research in India. But apart from investing in these transformative ideas, we also support research ecosystems to advance discovery and innovation to improve the health and well-being. We are funded by the Department of Biotechnology, Government of India, and the Wellcome Trust in UK. Apart from funding the research, cutting-edge research, we also fund various programs that promote various national and international collaborations. We promote professional skill building workshops and also organize programs on strengthening research ecosystem, and we promote science education, awareness and public engagement with science in India. You'll know more about our various activities and webinars that we have from time to time by visiting our social media handles that will be shown on the screen now. Now, before I start, let me tell you about certain workshops that we hold. So we periodically hold science communication and outreach workshops, as well as public engagement events throughout the year. To keep yourself updated on all of this research fellowships, research grants and events, kindly follow us on the social media handles that you can see on the below the screen. Or you can all go to any of these handles and type India Alliance. You will come across our posts. So more on India Alliance will be shared by my colleague Isha Goel towards the end of the session when she talks about funding opportunities available at India Alliance. So before we dive into the lecture, I will I have a few housekeeping matters to talk to you about. The first is that the webinar is being recorded. So at a later date, we will be posting this webinar in our YouTube channel. Your videos and microphones will remain switched off during the event to avoid any distractions. If you have any questions, you can always leave the questions for the speakers anytime during the webinar in the question box. There are separate question and answer sessions where we will ask you to ask the questions to the speakers. In that case, you can always raise your hand. You can see the raise hand option on your side or towards the lower side. So you will be able to see and raise the hand to ask the questions. Please make sure that the questions only relate to the topics being covered in today's webinar. In case you find the webinar ends abruptly due to any technical glitch, please join back using the same registration link that you have used now. Please also note that we'll be providing e-certificates to those of who attend the entire session and share a post-workshop evaluation with us. The certificates will be mailed to your registered email IDs within 6 to 7 working days. That means sometime during the coming week. We look forward to your feedback after the webinar, and if you plan on sharing highlights from the workshop through your social media handles, please feel free to tag us. Now we move to the agenda of the workshop. To the next slide, please. So the agenda for today's workshop is such that we begin with the introduction that we have already started. Then we have a welcome address, which will be followed by the first session today, the on genome editing in animals for enhancing productivity by Dr. Naresh Selokar. And a question and answer session will follow this, and then we'll have a short screen break of 5 minutes. Then my colleague will present the various funding opportunities at India Alliance, and we'll have the overall question and answer session at the end, and end with the closing remarks. So before we begin, I would like to call upon Dr. Ratan Chaudhary, who will give today's welcome address. To briefly introduce him today, Dr. Chaudhary is serving as an assistant professor at the College of Animal Biotechnology at GADVASU, Punjab. Dr. Chaudhary holds a BSc degree, a master's degree in veterinary sciences from IIT Roorkee, and a PhD in animal science from the University of Maryland, USA. He has 2 postdoctoral research experiences, one from the University of Kentucky, Kentucky, another from the University of Vermont, USA. The long-term goal of his research is to develop stem cell therapies for the dry period and mastitis in dairy animals. In addition to this, Dr. Chaudhary has been working on adult stem cell expansion and stem cell derived products for regenerative therapy in canines. He has several publications in esteemed journals and has won many prestigious awards for his work. With this introduction, I invite Dr. Ratan Chaudhary for his speech. Over to you.
Thank you. Thank you so much, dear Hima. So good morning to all. So it's my opportunity to introduce the topic. So we all know biotechnology is an exciting and ever-expanding field of science that harness cellular and biomolecular process of living beings like microbes, animals, plants to develop a usable product for mankind and society. The big aim of Department of Biotechnology, DBT, Government of India, is to make country a 100 billion bioeconomy by 2025. India has more than 200 biotechnology industry. The objective of continuing biotechnology education, popularly known as CBE Lecture Series, as Hema told, is to strengthen the biotechnological skills and provide updated information of biotechnology to the students, the staffs, researchers, and professionals. And number 2, to build a scientific network of students and professionals for their mutual benefits. The CBE provides learning of skill-based techniques, cutting-edge technology, equipment, and updated research-based knowledge of biotechnology. On behalf of College of Animal Biotechnology, Guru Angad Veterinary and Animal Sciences University, Ludhiana, IIT Roorkee, and India Alliance DBT Wellcome, I, Dr. Ratan Chaudhary, welcome you all to the 4th lecture of CBE on genome editing in animal for enhancing productivity. This talk will be on CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is a microbial immune adapted system to kill bacteriophage. Now this technology is being used along with Cas9, which is a nickase, and efficient RNA guided nucleases for genome engineering in eukaryotic cells with various DNA repair mechanisms. So in this lecture, Dr. Naresh will deliver the talk about the CRISPR-Cas9 based genome editing and in farm animals and their applications in enhancing animal productivity. I think he has been very actively working on this area in India, and it's my honor to we all listen him today. So thank you and have a wonderful day ahead. Now over to him. Thank you.
Thank you, Dr. Ratan, for your wonderful welcome address. And now we move to the first session for today. So, Abdul, can we have the slides up, please? So I'll begin before we begin the lecture on genome editing in animals for enhancing productivity, I would like to introduce you to Dr. Naresh Selokar. So let me briefly introduce him to you. He's a scientist at the ICAR National Dairy Research Institute in Karnal, India. He studied veterinary sciences at the Nagpur Veterinary College and earned his master's and a PhD degree in animal biotechnology from IIT NDRI Karnal. He has completed 6 months of Commonwealth Fellowship training in cattle embryology at the Ontario Veterinary College and 1 year postdoctoral training on CRISPR-based genome editing in mice embryos at the FLI Institute of Farm Animal Genetics from Germany. He contributed significantly to establishing a simplified, economical, and efficient handmade cloning (HMC) in buffalo and cow. His current research is to explore CRISPR tools for the production of more productive and healthier dairy animals. He has received several prestigious awards, including the Associate Fellowship 2020, Young Scientist Platinum Jubilee Awards 2019 from NASI India, Innovative Young Biotechnologist Award from DBT India, Young Scientist Award from SERB India, and Jawaharlal Nehru Award for Outstanding Doctoral Thesis Research in Agricultural and Allied Sciences by ICAR. So with this brief introduction, I now invite Dr. Naresh for his presentation and his talk today. Over to you, Dr. Naresh.
Hello, I'm audible?
Yes, you are.
Yeah. Thank you, Himanshi, for the introduction of mine. At the outset, I'm grateful to GADVASU University and particularly Professor Malik for giving this opportunity to me to deliver a talk or share what experience I have in terms of the genome editing in the country, as well as give an overview picture of genome editing in farm animals. I also thankful to Dr. Ratan for taking up an initiative in this direction to encourage young people to involve in the biotechnological research, and definitely through this lecture series, which the GADVASU and the IIT, they are jointly organizing with the help of the India Alliance Wellcome Trust. The more people will be divert their minds to join the biotechnological research in the country. And definitely, as Dr. Ratan has been mentioned, we have the vibrant biotechnology industry in the country. And in the future, we will be leading in the biotechnology research globally. I am also thankful to Himanshi for organizing this series of the lectures. And we have a couple of dry runs during my presentation today, before presentation of today. And thank you to her also for organizing everything on the platform. Today, so my slides are visible. Hello.
Yes, making a presentable mode.
So it is in presentation mode?
Yes.
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 Yeah. Now, so just a moment, there are you have asked to go. Now it is in presentation mode?
Yes, you are good to go.
But I have seen the video only. My slides is hiding behind the video.
Oh.
Now is okay?
Yes, we are able to see you.
Okay, so today, so I will go ahead. Anyway, so today I will talk about the genome editing in the farm animals for enhancing the productivity. I think Dr. Ratan has been just brief about what are the potentials of this genome editing, in particularly not in the farm animals, but in the other species as well as in human applications. So it is a very emerging field, and there are several applications, including the correction of the genetic diseases, diagnostic, treatment, and particularly as we are working in the farm animal institution. So we are exploring this tool to enhance the productivity of and dairy, particularly dairy animals, which are in the country. We have these buffaloes as well as the cattle. So why we need the genome editing? I think everybody knows that we have been going to be a most populous country soon. And what the challenges we have in the coming future is to feed this huge population that we have today, or maybe in coming days. And with the applications of these technologies, and what the technologies we will explore in the future is the genome editing tools, which include the GADVASU, TALEN, and the CRISPR, to provide the nutrition food to the people, as well as to protect the health of the people once the COVID has been emerges out in the today's scenario. So there has been a lot of concerns about the one health aspect. So there are these lot of diseases that have been transmitted from animals to the human and humans to animals. So we need also protect the health of the people, and to mitigate the challenges we have in the coming days or to be faced in the future, we need to explore the advanced techniques or these biotechnologies, and that one of them is the genome editing tool to mitigate these challenges. But what we have at the present in the country, where we stand in particularly in terms of the CRISPR research, if we talk about, so this technology, particularly for the manipulation of the mammalian genome, it has been initiated in 2012 or 2013. The first publication came from the United States, from the Feng Zheng lab and the Daduna and the Sharpinter. These two ladies last year has been awarded that Nobel Prize. So still, if you talked about the contribution or in terms of the CRISPR research, so United States has the more technical improvement or the involvement. What are the technologies we have today? It has been originated in the United States. Still, they have been forefront in these technological development. But in terms of because we are more, I and my organization, the Indian Council of Agriculture Research, they are more interested in agriculture application. So in terms of agriculture application, China is the forefront. They have been extremely or exploiting this potential of the CRISPR tool for the agriculture applications, which including the production of the productive crops and making the disease-resistant crops, animals, and so and so. So what we need, and then recently India also has been changed the guideline for the application of CRISPR research. I think 3 months back, they have excluded the type 1 and type 2 mutations from the regulatory authorities by the Department of Biotechnology and the Indian Council of Agriculture too. So what we need to invest in more, because this technology has the potential, and through this technology we can achieve the lot of targets we have been going to face in the future. So we need to invest more. In addition to that, more scientific workforce, so more people to be trained in this area, more students to be trained to boost the CRISPR research in the country. So if you talked about, because as I informed that there are these students also, or the young people, they're also joining. So my talk is basically, one part is on the basic aspects of the CRISPR research. And then the second part of my talk, I will focus on more about what we are doing at the NDRI. And then that is particularly on the buffalo, because we have been more interested to apply these CRISPR tools in the buffalo. So the first part, it will be the basic of the CRISPR technology, how it works and what is ongoing in the field. And that is particularly in terms of the farm animal. So if you talked about the CRISPR, genome editing tools, there are a couple of genome editing tools, which include the restriction enzyme, which is called as, I think everybody in the biotechnology, they use the restriction enzymes, and that is called as the original genome editor. The two protein-based genome editing tools, one is the GADVASU and the TALENs. They have been used from the couple of 20 years before, but their efficiency and the application in the laboratory is the costlier, so that's why they have not been widely applied to the many laboratories. When the CRISPR has been evolved from this bacterial immunity to the mammalian genome editing, it is very simplified and easily accessible, and any laboratory, very simple biotechnological laboratory, can use the CRISPR-Cas gene editing technology, and that's why it's considered as a revolutionized field in terms of the genome editing. So what actually these all the genome editors, what we have mentioned here, the 4 genome editors, what they do, they enable the changes in the DNA that leads to the change in the, because of a phenotype has been determined that the DNA, if you change any nucleotide there, it has been changing into the physical traits, and that ultimately everybody looking for. So these all the genome editor, the works looks like a scissors, so it has been cut at a specific site. There are the targeted messengers to be there, or targeted guides to be there, or target sequence to be there to recognize the sequence, and that allows the addition of any nucleotide or the replacement of the any nucleotide or the deletion of the any nucleotide at the DNA level. As for example, the sickle cell anemia, which is an genetic disorder because of the only point mutation, and that point mutation can be corrected. Similarly, in case of the farm animals, the bald, these are one of the disease, the CD18 disease that has been caused this genetic disorder in farm animal that can be replaced and corrected by the CRISPR tools. So how it has been discovered? So just a basic discovery of the CRISPR. So Francis Mozico, when I have an opportunity to meet him during my space visit at the University of Maryland. So when he was doing his postdoctoral research in 70s and 80s, that time he was not aware that what research has been done that 80s and 90s that has been very useful for today. So what actually the bacterial immunity, what happened when this bacteriophage is infected to the bacteria, the sum of the nucleotide sequences from the bacteriophage genome has been inserted into the bacteriophage genome into the bacteria genome, the bacterial chromosomal genome, and that is called the spacer. But when the second time infection happened, that time these are the spacer sequences, they transcribe into the guide RNA, and this guide with the Cas9 complex, they recognize these the second time infection of that same bacteriophage, and they have chopped off this bacteriophage genetic material, and that's way the bacteria make themselves against immunity against the particularly invading bacteriophages. And the later on 2012-13, the Feng-Zhang, Sharp-Pinter, and Daruna, they explore this bacterial immunity mechanism for the manipulation of the mammalian genome, and nowadays I think every laboratory and majority of biotechnological laboratory, they have been exploiting this CRISPR-Cas system for the various application, either knocking out, diagnostic, or the therapies purpose. So it's basically in the CRISPR-Cas system, we have the CRRNA, the CRISPR RNA, and the trans-activating RNA. What the trans-activating RNA do, it has been activate the Cas9 protein. It has Cas9 again the two domains, but it's a cleavage activating domain and the cleaved domain. When they have recognized with this 20 nucleotide, the sequence which is homology to the target sequence, and with the help of the PAM site, they recognize it and then make the double-stranded cut. When the double-stranded cut will be made, this is just an example of the in the embryos. So when they make a double-stranded cut, there are the two mechanisms initiated in normal repair pathway. The one pathway is the homology, non-homology directed repair pathway, that is called the NHEG. Another is homology directed repair pathway. What happened in terms of the non-homologous end-joining pathway, in that situation, there is a frequent cutting happen at the target site, and because of that, repair is not be done appropriately, and it is an error-prone process, and that lead to either the insertion of the some nucleotide or deletion of the some nucleotide, and that lead to the changing of the frameshift of the particular gene. When there is a frameshift has been changed, that lead to the either the unexpression of the particular gene or protein is not expressed. If it is expressed, there is a truncated and they're not able to perform the function. And that approach of the non-NHEG pathway has been used for production of knockout animals. Similarly, if your aim was to correct any kind of mutation, then you need to provide the donor template into the cell. When there is a homology directed repair pathway initiated, the donor template will be replaced the mutated one, and that lead to the transgene integration into the genome, and subsequently that mutation to be corrected. So in terms of the application in several species, I think everybody aware that it has been wider application in terms of the crops, in terms of the animals, in terms of the humans, and in terms of the diagnostics. So if you talked about more about the crops and animals, so it has been several works has been ongoing in the country too. The Indian Council has been leading the CRISPR research at the couple of institutions. They are working on the development of the improved varieties of the wheat, rice, and the tomatoes, and the bananas, and so on and so forth, agriculture. In terms of, if you talked about the animal science or the farm animals, the NDRI has been working on it, and a couple of more institutions, CIRB, Coast Kashmir, as well as the CIRG and the National Institute of Animal Biotechnology, the NIB. The couple of institutions working, exploring this CRISPR tools, production of more productive animals. What we do at NDRI, we are working to improve the meat yield of the dairy animals, particularly buffalo, because the buffalo is not only the dairy animal, it is also a meat animal. In addition to that, we are working on the production of the disease-resistant buffalo, which particularly for the manipulating the CD18 gene, and a couple of the basic research to understand the development of biology in embryos. For example, why the significance of the ox, sox, and an owl, and so on and so forth. Similarly, in terms of if you look at it, the other the research area, for example, correction of hemoglobinopathies, very commonly they have been exploring for the hemoglobinopathies, mean thalassemia, sickle cell anemia, and so on. So antibiotic resistance also one of the concerns today. The people they are exploring to find an alternative to the antibiotics. In embryos manipulation, particularly human, but is not legally allowed. You might be heard about this attempt has been made by the He from China, and there are a lot of controversies. And now there have been regulation that you cannot do the germline editing in human embryos. But definitely there are some issues to be taken care, and the legal issues is ongoing in terms of the human embryo germline editing. Now coming into the Indian context, as my profession as well as my responsibilities to the country or to my organization, we know that in case of the farm animal contribution is this in this country, or for the providing the good food from the farm animals means in terms of quality milk, as well as not only the quality milk. If you look at these, we also need these meat product from the animal, particularly from the poultry or the fishes and so on and so. So what technologies we have been using for improving the productivity of the farm animals? When I talk about the productivity of farm animals, include both the milk and the meat. So from the many generations of the years, we use the selective breeding, in which what happened in the selective breeding, what we do, in which we the elite nature of an animals, or particularly semen from the elite animals, to be used for the artificial insemination and to try to upgrade the next generation animal with the higher productivity. For example, in case of the sheep and goat, their target was to enhance the productivity in terms for the milk purpose. And in case of the dairy animals, cattle and buffaloes, so the bulls from the dam, which is producing higher milk, they have been used to produce the next generation offspring or the daughters, which has been producing the higher productivity. And that's why today, because of the selective breeding, the country has been the top rank in terms of the milk production, as well as substantial rank has been improved in terms of the meat production too. And that's why we have the poultry industry has been developed in the country. And recently, the genomic selection now coming up to select the animal for the higher productivity or the higher genetic merits. So this one approach, this has been the country has been exploring to improve the productivity of the farm animals. In addition to that, what the most important another aspect is that the nutritional and management intervention, that is also really an important aspect. Still, we in the country, we have the very small holder farmers, and to provide them appropriate nutrition to this particular animal to maintain the sustain or to maintain or continue to sustain the productivity of these animals, the nutritional intervention, lot of the organizations of the ICR, State University, they are working on the nutritional intervention to provide the package of practices of the management and nutrition to maintain the productivity of animal, or what kind of the packages to be required when we produce the higher productive animal. That we have been doing from the last decades or so. We have been working on that. And what now we have the new biotechnological tools for enhancing the productivity of the animal, or to make the animal to be a disease resistant or treatment of the disease. For example, now the government has been initiated these one biotechnological tool. I can say it is an OPU technology to produce the elite nature of elite animals, particularly for the dairy animals, and that is particularly toward the cattle. So that if you have the more number of the elite animals we have, so our productivity will maintain, as well as if your productivity will be maintained, as well as the number of animals required to achieve that productivity or to achieve that production will be lesser. So aim was to reduce the animal number, but in the same time we have to continuously provide what the requirement in terms of the milk as well as in terms of the milk. So what I do in my laboratory here at the NDRI, we use the biotechnological tools, which include these embryo transfer, animal cloning, and now we have been exploring this. This is genome editing for the increasing productivity, and particularly both targeting milk as well as the meat. So why we need to edit the farm animals? I think for improved trade, improved health, we already discussed animal welfare too, and the human health, because ultimately we are all together, all the biosphere we have, it is in one common health. So we need to protect all this aspect from the animal point of view also, their welfare point of view also, and the human health too. Now, why we need to editing in the farm animals? So in terms of the animal production system, we do the regular breeding of the farm animals from the generations. So normally what happens, the unfavorable animal population, when we do the breedings of the unfavorable population with the favorable means higher productive animal or the genetically superior animal. So this is a classical, the upper one is a classical breeding plan. So it takes an 8 to 10 generations to upgrade a desirable trait, but that is also at not that extent. So it may be up to 20%, 30%, or 60, or maximum of 8 to 10 generation, we can achieve the 70 to 80% of that particular trait. And that is the drawback of that particular classical breeding program. It takes then years, at least 8 to 10 years in case of the dairy animals, if you talk about the cattle and buffaloes. What we added advantages in terms of the new tools, which is called the genome editing tools, which include the Genome Editing and CRISPR. So within these one generations, you can achieve a favorable trait which you have been expecting in particular animal type, either for the milk production trait, or either for the meat production trait, or either for the disease resistant trait. So because we have been manipulating the genome at the very early stage, at the embryonic stage, either you can manipulate genome at the somatic cell, or you can manipulate the genome at a very early embryo stage, and that lead to the development of and ultimately to the desired animal at the first generation itself. These are some of these selective examples globally. So there are a lot of research has been happening in case of the farm animal, because they're not much highlighted in terms of the social media or these print media. But in scientifically, if we talk, there are a lot of happening in terms of farm animal. So very famous, these are some famous examples of these the genome editing applications in the farm animal. The hornless cattle, it has been introduced in the United States. So the cattle, when they born, they don't have the horn. So it is a welfare point of time, welfare aspect, they produce the hornless cattle. That's for the pole locus they have integrated into the genome. Another famous example is the resistance against the bovine TB. I think many of the people who has attended, they understand, they may know that the government of India has wanted to eradicate the tuberculosis from the country by 2030, but without targeting these animal population, because tuberculosis is a genetic disease. Still, India, I see in the many in my villages also a lot of people they infected with the TB, and majority of the time the TB is not the human tuberculosis; it is called the bovine tuberculosis. So animal can harbor the human tuberculosis bacteria, and human can also harbor the bovine tuberculosis bacteria, and the symptoms they are similar. So we need to target this tuberculosis also in terms of the farm animals to eradicate the tuberculosis from the country. And in that situation, the target gene, which this has been reported from the China, they targeted this NRNPA gene from the macrophages, because the tuberculosis is they are multiplied in the macrophages. Another example of the CD163 gene, the pigs. These are the deadly disease in pigs, the porcine reproductive and respiratory syndrome virus. So they have been now patented, and they wanted to commercialize it for the application in both in the United Kingdom and the United States. They produce, and the other countries too also they produce these resistant pigs. Another example is in case of the ruminants, the increasing muscle mass. So myostatin gene has been edited in cattle, in case of the sheep, goat, and the pigs, and the several species has been produced or improve the meat productivity. Another is for understanding the reproduction aspect, the germline ablation, the nanos2 gene has been knocking out, and they understand that this gene is really important for the reproduction aspect in the pig, and probably it will be applied to the other species. In addition to that, there are the several research ongoing for the genome transplantation, for making of these with the animals for the more yield of the fibers in case of the sheep and sheeps. How the quality of the fibers to be improved, and these are the couple of examples, which is examples available and published by the scientific community to demonstrate that this technology, the CRISPR technology, has a potential to produce the more productive as well as the disease-resistant animal. Now coming into the next part of my talk, what we have been doing in Buffalo at our institution here at the NDRI, as well as my collaborators from the other organizations. So I will just highlight the importance of Buffalo and then to the shift to the genome editing aspect. So if we just an overview picture of the Buffalo, what are the Buffalo contribution? Many people they don't know how the Buffalo has been contributed, particularly from the non-veterinary background or non-animal science background. So if you look at the global population, India boasts around the 96% of the entire world population. Whatever the buffalo we have in the entire world, the majority of buffalo population they have in the Asian countries, mainly India, China, and the Pakistan. Out of that is around, I think, 60% they have been in the India itself, and that Indian buffaloes are the higher productive in terms of the milk as well as in terms of the meat too. And if you're the global share in terms of the buffalo milk, if you talked about the 15% of global milk production, it has been shared by the buffalo, of which in Asia, if we talked about, is a 35%. And when we talked about the India, whatever the milk produced, as on today, if you talk about the 210 metric million ton of milk produced in the country, out of that is around the 50%. The half of the milk produced in the country has been contributed by buffalo. Despite that, the population of buffalo is half of that of the cattle. So there is a buffalo contribution in terms of the milk is in tremendous is there. In addition to that, there are a tremendous contribution of buffalo in terms of the meat industry. So every year, if we talk about the Indian meat industry and exporting the meat from the buffalo and its product are 20,000 crore rupees, and which is around the 4.2 million tons of the meat annually to the mostly Arabian countries. So buffalo is as a multi-purpose animal. It's called a black beauty of our country, and it has produced the milk, meat, and work power. And so because in many, if you go to the villages level, you can see a cart which carrying the ganna or sugarcane or these hays from the farms to their homes by the buffalo cart. So you can easily observe in the countryside. So again, in terms of the buffalo, what the species we have, there are the two types of the species. One is a riverine type, which has a 50 chromosome and called the dairy purpose animal. So for example, the Murra, Neeli Ravi, which is famous in Punjab, and these Jafrabadi and Nagpurian. So there are around the 19 buffalo breeds of this beautiful species. And then another type, which is swamp type, which is particularly located in the northeastern part of the country. They are low milk producers, but they're mainly used for the meat purpose, and that has the 48 chromosome. What the famous breed in terms of the Indian famous breed is the Murra, which is globally accepted. Another couple of breeds from the other country too, because their economy, particularly agriculture-based economy, is also dependent on buffalo. For example, the Pakistan, they have the Ravi buffalo, which is similar to our Neeli Ravi buffalo, and Mediterranean buffalo in the country. So this species has a significant role not only in India. I think we must acknowledge the contribution of buffalo for its agricultural development in the country as well as the dairy industry growth. But in addition to that, the other countries also nowadays, the African countries or European countries and Latin American countries, they are interested to adopt the buffalo as an industry there too, and it's very growing fast. Now, what the methods we have been using for the genome editing buffalo? So to have to produce a live animal, it's a challenging task. So you can produce an somatic cells in the laboratory, you can produce an embryos in the laboratory, but ultimately reach to produce and calf, you should have a strong infrastructure as well as strong, you can say the manpower or resource person or the skilled person to do the embryo transfer, to do the manipulation of embryos and ultimately lead to the production of the calf. So what we do in our laboratory, we worked on the SCNT from last 20 years at the NDRI, and several clones have been produced in the NDRI and another institution at present at the CIRB. So what we do in the cloning process, it is a basic process that will be also explored for the production of genome editing calves. So what do we, with the somatic cells from the any individual which we wanted to edit, individual means a buffalo, either it's a male or female, so we need to establish the somatic cell. At the same time, we brought the ovaries from the slaughterhouse and then oocyte to be matured in the laboratory, make them enucleated them manually, but the method we develop at NDRI is handmade cloning, and then fusion of the two enucleated sites with the one somatic cell, and then we need to activate them and culture these embryos, whatever generated, for the 7 days in the laboratory in the control incubation, and then transfer into the surrogate animal, which has been same synchrony that of the embryo stage. And once we have been establishing the pregnancy, we need to examine to the ultrasonography and then care at the time of the birth, and then ultimately lead to the production of the cows, which has been genetically identical to that of the from whom we took the somatic cells, or from whom which somatic cell has been edited. So these are the basic method to be used for the either for the clone animal production or for the genome edited animal production. So just I would like to highlight the journey of the buffalo cloning in the country, because this is important, because to produce a genome edited animal, what approach we are using here at the NDRI, we use the SCNP based genome edited animal production, because this technique is well established with us. So what we achieve in terms of the buffalo cloning, the first clone in the country, or we can say the first clone of the world of the buffalo, which is the Riverine buffalo or Murrah breed, in 2009 at NDRI. And at the present, through this lot of modification, what we achieve, our embryo production rate is around the 30 to 35%. But the other challenges we have, despite having the similar embryo production rate, that of the IVF method, but we have the live birth rate is less, around 6 to 8%. That's the challenges we have. Normally, in case of IVF embryos, it gets around the 30 to 35%, and the artificial insemination is around the 50%. But what the other, in addition to poor live birth rate, we also have a challenge to minimize the losses of the early pregnancy. We have around the 25 to 30% of the whatever the pregnancy we established has been lost during the gestation period. So this what we have been achieved. We are working on this direction to how to improve the live birth rate, as well as how to minimize the losses of an early pregnancy. But what we have achieved as on today, by 2021 or 20, now it's this bit number has been increased, but it's a data of December ended 2021. We have around the 21 clone buffaloes, of which the 16 are the breeding bulls, because the Indian Council of Agriculture Research are interested to produce the elite breeding bulls, so that the semen to be used for the faster multiplication of germplasm, because one bull can be produce the lacs of the daughter, and that has been faster way of genetic improvement. So there is any bull which has been top ranked, so we have cloned them, a couple of clones we have top rank at NDRI as well as at the CIRB. And recently the CIRB has produced the 7 clone copies from the single breeding bull. So one bull, the 7 copies has been produced, and again we proves that the recloning in the buffalo can also be possible, and reclone calf was introduced. So through the year of research, what we have, because we are asked so many questions about why this is really normal or not. So we conducted a couple of studies, and that has been published in the scientific journals. So what we observed, that once the clones have been attained the maturity, we do agree that there are the early pregnancy losses are there, maybe early calf mortality is there, but once the clone attain the maturity of an age, they have the normal growth. Those clones, they have attained the maturity, they have the normal growth, they have the normal health, their fertility and production. We also use the semen from the clone bulls and produce these around the 100 to 200 of their offspring. And now we have been recording the production from the clone animals as well as from their progenies, their daughters, and the semen quality from their sons. So the clone bulls and their progenies too, we have been working on it. So what the data we have as on today, that once the clone attain the maturity, they have absolutely normal lifespan. We have at NDRI, the clone which is born in 2010, so almost 12 years of buffalo is here at NDRI, which is an oldest buffalo we have here. This is just an achievement which has been highlighted by Indian Council of Agricultural Research. Multiple copies from the one bull, which is M29, is a very famous bull in Haryana, and this bull has been used extensively for breeding purpose. What our target was to, if we have multiple copies, so these bulls can be given to other part of the country, for example, Andhra Pradesh, Bihar, Uttar Pradesh, or maybe a southern part of the country. So their semen can be used to disseminate to other part of the country for the genetic improvement program. And the lower side, this one is this reclone of an earlier clone bull at the Hisar. Now coming into, this is a bit about, I will not go into the detail about the more scientific aspect what you have been done in terms of the transcriptomics, genomics, and these and of these clone animals, what you have been done, or some other aspect, just gives the glimpse about the what you have been achieved as on today in terms of the cloning process, because we are going to use this cloning process for the production of genome edited animals. Now, global picture of an buffalo genome editing, there are a couple of studies and published from the IVRI, Dr. Sarkar lab has been worked on it, and from China, because these two countries, they are majorly working on the buffalo productivity enhancement. So a couple of studies has been used to application of the CRISPR, and they have been in culture system or in vitro system, and they identified the, for example, Dr. Sarkar lab at IVRI, they understand the role of the corpus luteum during the reproduction, and the couple of genes they are knocking out, and they identified that what is the significance of that particular gene during the corpus luteum formation or corpus luteum function. Similarly, from the China, they use the myostatin gene for the knocking out to in case of buffalo, and just for the optimization experiment, as well as these long of the Y chromosome, the DDX3Y gene, and how it has been able to produce the sex embryos. So a couple of studies and very recent studies published from India as well as from the China. What we do at our laboratory, we worked on these two important genes, which I'm sharing today. In addition to that, we also recently initiated a couple of more projects in this direction. But what I share today is the knocking out and production of these animal for the improved, in addition to the milk, for the improved meat of the myostatin gene knocking out and the knocking at the CD18 gene. This CD18 gene is important in terms of the this disease which has been buffalo infected, for example, Anemia hemolytica. What the information we have as on today, we don't have any live genome edited buffalo produced yet. So our challenge was to produce the live animals to demonstrate that this technology is physical in buffalo too, as it has also been demonstrated in other species, the cattle, sheep, and goat from the Western world. This project we have been working in collaboration with the CIRB, CIRG, and COS Kashmir. So NDRI is the lead center in this project. What we have been trying to do in this project, we have been three species we selected. There's a buffalo, sheep, and the goat. All the three species are very important in terms of the providing the meat to the one buffalo is for the export value, and the sheep and goat for the in-house consumption. So we selected the 4 individuals from this buffalo, sheep, and goat. We have already characterized the complete myostatin gene from this species, and we are under process of publication to this characterization of the gene. And then now we need to we are in process of optimization of the pipeline for the editing in the somatic cells as well as embryos of all the 3 species. And then we have a couple of cell lines we generated, edited cell lines, and these edited cell lines, as per technical program, it will be used to produce then edited embryos. At the same time, we also initiated the edited the microinjection in the IVF zygotes of these, particularly in our laboratory in buffaloes. Once we have achieved to produce an embryos, it will be, in case of the non-surgical transfer, will be done in goats and the sheep at the respective institute. The CIRG, it's again the ICR institution, Central Institute of Research on Goat. They are working on the goat aspect of this gene, and COS Kashmir, they are working on the sheep aspect of the genome editing of that. Once we have been established pregnancy in produced animal, we need to assess the growth, health, and performance of this treatment, how they're performing well or not. So this is an overview technical plan of this our project we are working jointly with the other institute. Now coming into the why we choose this myostatin gene. This is very important in terms of the regulatory point of view also. So myostatin gene is a negative regulator. When there is a higher amount of myostatin available in the muscles, then the growth is lower. When there is no myostatin, then there will be the higher growth. And very interesting is that these are the naturally mutated farm animals. For example, if you see here the Belgian blue cattle, and this is a naturally mutated myostatin cattle breed is available, and these breeds are already in a food chain. Food chain means they are used for the food consumption. So their meat has been used, the beef has been used in the West, in Europe and the USA for the food consumption. Similarly, Belgian, there's a Taxel sheep breeds and Piemontese cattle. They have the natural variant or naturally mutated animal. So in terms of the regulatory point of view, so if we artificially mutated also, we assume that because of the new guideline, if there is a point mutations of any particular, and every the natural variant is available, it's easy to get a regulatory approval. That's why we chosen this this gene, and it has been well proven gene in other species too. And through the research, it has been proved that not in particularly in the buffalo, but in other species, sheep, goat, pig, and cattle, is that if you artificially knocking out of the myostatin gene, it's also been improve the meat yield of the particular animal, and it shows the double muscle mass phenotype, the meat yield being increased. Then again, it has been proved that both the naturally as well as artificially mutated animals, they grow well, healthy, and fit to reproduce. This is very important. Even if you do the mutation artificially in these animals, they are able to produce their progenies, and they are absolutely perfect, and their yield is also increased. So keeping this in mind, we choose this myostatin gene. Now coming into the Thickening program, so this species we selected, genomic DNA amplified, characterized, and the chop software has been used to design in the CRISPR-Cas complexes as well as CRISPR-Cas guides to clone into 2 vectors. We use TX45, TX49, and then everything has been done in the screen now, and they have been used for the transfection into the buffalo fibula cell. This I'm presenting the buffalo data only, not the sheep and goat data. So this what we have been optimized is that what are the conditions are required to transfect the buffalo cells with the larger vector, which has the CRISPR components in it. And this vector, CRISPR vector, is at around this 9 kb vector. And when we worked on the smaller vector, we also worked on the transposon-based transgenesis for the optimization strategy, and we found there are excellent transfection rate we achieved around 80%. But with the when we use this vector, we are getting a difficulty in the transfection. So we thought to optimize the conditions to achieve the maximum transfection. But despite of a lot of efforts we put, with a lot of combinations we put up the electroporations, so they're different duet size and different amount of plasmid. But what we achieved till today, we have maximum transfection rate is 15% only. That is the limitation. But despite that, when we go for the puromycin selection and done this screening, that really editing happening is not, despite having the lower transfection rate, we could able to achieve the editing in the somatic cell. This is just an assay to confirmatory assay that editing has been happened. This is the lower band, which is representing the editing event. And then we again confirm that what kind of the edits are happening at the population of the cell. This is sequencing of the PCR fragments, and it has been shown from the 1 nucleotide deletion to maximum we achieve as the 45 nucleotide EDs, and some of them have the medium size ED. So this we have achieved, and we when done. Now the next target was to, when we do have editing of the population in the cell, now we wanted to generate the clonal proliferation. So we again optimizing the we have optimized the conditions for the generation of single cell lines in a buffalo. So we use the different surfaces, different medias, and different combination of the growth factor and so on. So now we have developed a method for generation of the single cell. So it's very simple method, but with the combination of the growth factors or other which are required for proliferation of the buffalo cells, and that able to generate from the single cell the entire colony of the cells, or a lot of population of cells has been generated. And what we have as on today, our attachment rate, when you see the one cell in the 96 well plate, it is around the 30 to 40%. Out of that, these 20-25% of the population, they have proliferated and generated into the multiple populations, means it can be reached to the T25, and then subsequently it has been cryopreserved for the subsequent use. This also we optimize in our laboratory here. And then our next target, whatever the colonies we have generated, these are just one example experimental data I'm sharing here. So a couple of colonies have been generated from the single cells, and then our next target was to confirm that is there really what kind of the mutations what we have been intended has been happening or not. These are the 9 colonies. We generate colony, we label as a colony number 1. If you, the red highlighted colony is number 6 and 7. On the basis of the T7 assay, it is demonstrated, we can predict that there is the editing event happen, but not exactly which nucleotide has been changed, or either is it deleted or is an addition. We need to reconfirm it again by the scientific. I will show in my second slide. In addition to that, we, as I mentioned, that we also worked on the transposon-based transfection methods to optimize the transgenic embryo production, and it has been well established in our laboratory because the smaller vector size, as I mentioned, that around the 80% of transfection has been achieved in case of somatic cell, and easy to generate the single cell line. So these are these clone embryos which are expressing the green fluorescent protein. So This method we want to optimize, and the similar method we are going to use for the production of the clone embryos. This what the optimized method we have developed. In addition to that, we at present we have the single cell 5 colonies and their clone embryos introduced. So this is a similar line in the direction to produce of the transgenic animal. Now, whatever the colonies we have generated in terms of at that 8 colonies, so they have to again need to screen that what kind of the edits they have, either it is a monoallelic editing or the biallelic editing. Then we screen for the Sanger sequencing and confirm that the colony number 8 has the monoallelic editing of a -8 edits. -8 edit means one of the allele of the myostatin gene has been knocking out for the 8 nucleotide. Another allele, for example, colony number 7 has the -3 edit, which means is that 3 nucleotide, here is the 3 nucleotide has been deleted from the myostatin exon 1. What are the rest of the colonies we have been sent for the screening? For example, colony 1, 2, 3, 4, 5, they are found to be non-edited. So we will not go with this one, and we will go with this colony number 8 and colony number 7. And one more colony, that is a colony number 6, which is really interesting. It has been biallelic editing, and is one of the allele has been edited for the 7 nucleotide. Another allele is has editing for the -3 nucleotide, means 3 nucleotide deletion, and the one allele for 7 nucleotide deletion. What works we have been continuing now. This is the vector-based approach we have been used for the initial optimization, experiment, and generation of single cell lines. So we are screening the cell lines for the off-target, because that also need to be shown. If the animal produce, then without showing them to have any unintended off-target effect is not been recommended to. We are exploring that. We are also utilizing this edited cell lines for production of the embryos, and then subsequently these embryos will be transferred to the recipient animal. Since in buffalo it has a breeding season, which is started in the October and the onwards until the February. So during that time, I think in coming this breeding season, we will transfer these embryos for the establishment of the pregnancy. We have everything has been checked in terms of the cell, and then I think we are ready for the embryo transfer in this coming breeding season. In addition to that, now because CRISPR has been very evolving technology, so now we are shifted from the vector-based strategy to the RNP strategies. So we have also initiated this RNP strategy with the Lonza nucleofactor. So now previously we have been working on the newborn animals or fetal fibrillas. Now we are shifted to the adult animal. And these adult animal cells, for example, we choose this 2501. Why we choose this 2501 bull? Because we already have the clone copies of that, and that will be considered to be our control animal because we are using the cloning technique. So these cells will establish, we electroporated, and then confirmation again of with the T7E assay and going for single cell culture. And now we have generated the couple of colonies from this, which is at present we have 18 colonies. We are screening them for the monoallelic, biallelic, and in the coming breeding season we will explore them for the embryo production and then transfer into the recipient animal to try to get in pregnancies, and hope if it is everything goes well, so in a couple of maybe a year and so we could able to produce the edited animals, particularly for Murrah breed having these improved meat yield too, in addition to the milk yield. So we are working on this direction. In addition to that, we at NDRI we have a goat also. So in the collaboration with the CIRG, we also initiated this CRISPR editing in the goat cell. So we brought these 2 goat samples we use, and then we electroporated again at the similar principle with the RNP strategies, and then confirmation again with the T7E assay and generation of the single cell colonies. What we observe in case of the goat, the culture of goat cell is easy. What we observe in our laboratory. So if we remember the data of this buffalo, we attachment rate and the proliferation rate poor side is around the 30-40%, 25. But in case of the goat is very, very higher. So they are easy to culture in the laboratory, and that's why we are able to generate here the 34 single cell colonies. And these colonies has been, now we are screening, and since it has been these colonies we already shared with our partner institution to produce an embryos and transfer at the CIRG, and hopefully they have been attended to production of embryos and transfer at their institute in Mathura, and they will try to establish the pregnancy. So this work is already initiated there, and we are looking forward to have establishment of the pregnancy. This is a bit about this overview of this Myostatin project, which is working at the NDRI and the partner institution. What the other project we worked here This is a really interesting project on the CD18 project. It is an Indo-US grant. So when I was previously, I was at the CRB there. I have initiated this project. Now it has been same continuing here at the NDRI. So CD18 gene has an important role in terms of the there's a genetic disease also, for example, BALD, and another important disease which has been lead to the infection in the dairy animals, either it is cattle or buffaloes. There is a Mannemia hemolytica. What happened in terms of the pathogenesis has been well established of the Mannemia hemolytica. The toxin secreted by this Mannemia hemolytica bacteria, they have bind to the CD18 receptor. They're very interesting to note that the CD18 signal peptide are intact in the ruminant species. Ruminant means the cattle, buffaloes, sheep, and goats. They have the intact signal peptide, and because of that, whatever the toxins secreted by this Mannemia hemolytica is a bind, and that will cause to the cytolysis of this leukocyte, because CD18 is expressed on the leukocyte. This bacteria is a common cells in the upper respiratory tract. Whenever there is any kind of stress, either is in housing, temperature, transportation, and weaning. And if we talked about the Indian scenario, our animals, they are in the stress of the weather, their housing, and sometimes the transportation also. So there are a lot of incidences I also found in the couple of the farms available in the Haryana. They have been reported with the infection of these Mannemia hemolytica, and it's caused the death to the high rate of the mortality in the particularly young calves. So keeping in this mind, so we thought to propose a concept about the production of the resistance buffalo, which has been CD18 point mutation, so that the signal peptide is not been available or not been intact on the the leukocyte. So whenever there is even if there is infection, they will not cause the cytolysis of the cell. And this was a grant we do. What we have been done on this project, we have screened the two breeds of the buffaloes. One is the Nili Ravi and another is this Murrah breed. We amplified, sequence, and present is that here, if you see that this is a published paper in the by other authors, they have shows that in this particular ruminant species, and they have the cleavage inhibiting amino acid, this glutamic acid, at the -5 position. And in case of the non-ruminant, they don't have this cleavage inhibiting amino acid, and because of the 1-point mutation. So in case of our animals which have been targeted, we also found that our animals, means buffalo also, which we have been in the country, Murrah and Nili Ravi, they also have the cleavage inhibiting that amino acid, the glutamic acid, at the -5 position. So we wanted to replace it. So to have that, we have been to optimize, because this is a knock-in approach. So we need to optimize all these transcription conditions, the parameters, and then really whatever the guides we are designing, they're working well or not. But what we have today, we generated a couple of colonies. If you look at here, the sequencing data of the colony 1, colony 2, and the colony number 4, they have an editing event at the CD18 signal peptide receptor. So this we have been achieved, and now again we have been exploring this, continuing this aspect to produce these embryos and then transfer to the coming breeding season. So these are just chromatograph data of this confirmation of monoallelic and biallelic editing of this couple of both. They have the biallelic editing and other have the monoallelic editing, and through the another approach, I think the GenNet approach, the publication is already published in terms of the cattle. So they demonstrate that the Mannemia hemolytica resistance cattle they have been introduced to the GenNet approach. So we are exploring the CRISPR-Cas to production of the CD18 resistant, or CD18 means Mannemia hemolytica leukotoxin resistance cattle, not a cattle, a buffalo in the country, and we are working on this direction, and we have a couple of leads in it, and we continue our attempt to produce the live model animals. So now I sum up talk of my talk is that we are working our laboratory, we working on the myostatin CD18 gene, which is under progress. So we have good leads in this direction, and we continue our code. In addition to that, we are also been exploring some of the basic research to understand the roles of the OX, SOX, and ANOG in early embryonic development. That is one of my postdoctoral fellow is working on that aspect. Recently, we also have been exploring the hypoallergic milk and then unsaturated fatty acid in milk, but that is not been initiated. We have a project just been approved a couple of days back, and then we will initiate on that aspect. So what we understand in case of the farm animal, I would convey to that any trait, either it is a production trait, health trait, or the diagnostic trait, can be edited by or can be edited or manipulated by the CRISPR technology. And what we have in the country as on today, we have very well established buffalo cloning technology. I think this is in addition to the OPU IVF, another advanced technology we have in the country. As far as any of the laboratory in the world, we established the buffalo cloning technology in the country, and it's very well established. The technology now, the technology is mature well to translate it into these other laboratories. I know in the GADVASU also, Dr. Narendra is there. He's working on very excellently on the OPU IVF, and they produced a couple of several the progenies of through the OPU IVF, and hope in the coming future also they can initiate the work of the cloning too. What we have highest possibilities in buffaloes. There are a lot of possibilities and opportunity in buffaloes because it's very important species. But in addition to that, we have lot of challenges to working with the large domestic animal, particularly for the buffalo, the breeding cycle, the showing of the stress behavior, the transfer conception rate, and their cells in the laboratory culture. They are also a challenge, but despite that, we have an highest opportunity to explore the genome editing tools in buffalo. So I would like to acknowledge for the generous funding from the ICAR. Without the funding, this kind of research cannot be possible. ICAR has been so generous to us. They have been regularly funding from last 10 years at least. When I joined the ICAR organization, they have been providing funding in this direction, either buffalo cloning, OPU, IVF, stem cell biology, as well as now the genome editing, the generous funding to us. So thanks to the ICAR. In addition to that, Department of Biotechnology also has generously supported us a lot of projects in terms of the particularly OPU IVF and the cloning. And one of the project, my DBT project, has been first project which has been approved to me, I think 3-4 years back by the DBT on the genome editing, I think, and that's where we initiated the genome editing in our laboratory. Also the IUSSPF, the Indo-US Science Technology Forum, which has been supporting the CD18 project. So I acknowledge them too also, as well as my two organizations. My first organization, which I joined the service in the council, Central Institute of Research in Buffalo, and there are a lot of dynamic leadership that time there, and still today they are continuing that legacy from the CIRB. And the current organization and my alma mater, which I've always desired to be here, and I wish to be here, and then the opportunity is given to me to work for the NDRI, and thanks to the leadership at ICR as well as NDRI to be here, and then from last 1.5 years I'm working here and taking care of all the assisted reproduction technology at the NDRI. Our team at NDRI, my mentor Dr. Chauhan is still here at the NDRI, and my colleague Dr. Sachinanda Day, as well as Dr. M.K. Singh, and particularly the student I would like to acknowledge who worked on the Myostatin project, Bosco, the Devika, and the Raju. They're trying to develop these knockout cell lines for the Myostatin, and for the CD18 project, my PhD student Kamlesh, Seema, and the Sonu, they worked on the CD18 project. The team, my previous team at CRB, Dr. Yadav, Dr. Sharma, and Dr. Kumar, and Miti, still they are continuing the activities of the genome editing project as well as buffalo cloning project there. Also, I would like to acknowledge it to them too. And once again, thank you very much for the India Alliance opportunity and the GADVASU for giving this opportunity to share my laboratory work on the genome editing aspect as well as the overview of the genome editing farm animal. Thank you once again. Thanks. Hello.
No, yeah, thank you, Dr. Naresh, for such a.
So I think I have been on time.
Yes, of course. So I just want to know if any of the attendees have any questions or anything they want to ask about the topic they have heard about. So they can raise their hands and ask the question. We'll unmute them. Or even the questions can be written. Okay, I can see question. Just a second.
I think I have a quick question, if I am allowed.
Yes, sure.
So thank you so much, Dr. Naresh. This was a wonderful presentation, and I'm glad to know that India is doing great, especially under CIRB, and the entire team of yours is doing a wonderful job. So since my field is on stem cells, so I would like to know a little bit about how the manipulation of stem cell using CRISPR can be utilized for production efficiency or increasing production in animals. Any thought on that?
Yeah. Hello, audible? Yeah.
Yes.
Thank you, Ratan. Yeah, it's I think if we talk about the stem cell, that when we use a stem cell as a donor to produce an animal is an efficient in terms of the pregnancy rate as well as the less abnormality. Either it is a mesenchymal stem cell or the embryonic stem cell or iPSC, it is easy. In terms of manipulation, absolutely there is a no difference, or I must say it's an easy in terms of the stem cell for doing the manipulation through the CRISPR, particularly mesenchymal stem cell. But only challenges in terms of the stem cell because it has an investment in terms of the culturing, and that is particularly for the iPSCs or the embryonic stem cell. But for mesenchymal stem cell, it can be very easy, and you can manipulate the stem cell, mesenchymal stem cell, and use as a therapy purpose, or you can insert any desirable gene there, or you can knocking out any unwanted gene and then insert transfer that mesenchymal stem cell for the therapy purpose. I know you are working on the Myostatin, so it can be a very good tool for application of that mesenchymal manipulated stem cell for the regenerative therapy to cure the mastitis, as well as other cases of the wounds and other things. It's not that much difficult. It's an easy task.
Okay. Thank you. Thank you so much.
Thank you. I see a hand up, Venkatesh KM. So I'll just unmute them so that they can ask their question.
Hello, good morning, sir.
Good morning.
Sir, as I can see, most of the economic traits are polygenic in nature. Is there any validated study that only MCN or MSTN knockout gene can produce normal muscle growth, good muscle growth as compared to the knock-in gene?
For the muscle growth, you are asking that only knocking out of the myostatin is sufficient, or you need to use the knocking?
Yeah, because most of the economic traits are polygenic in nature.
Now, economic traits, if we talked about the dairy milk trait, is polygenic in nature. In meat, if we talked about, there are a lot of studies has been done in terms of the meat, and this is only a single gene which is governing 80% of the muscle growth is governed by this gene. So it is well established. If you remember clearly during my talk, I mentioned is that there are natural variant of the myostatin knockout. There is a pedomentacea cattle or Texel sheep or Belgian blue cattle. Because of this gene mutation, they are showing the muscle growth is a huge muscle growth. And the first they are demonstrated in case of mice, and that is why it is called as the double muscle mass phenotype. So there is absolutely no problem if you're knocking out the single gene also, it will improve the muscle growth by 30%, 40% in terms of the farm animal. But definitely in case of the dairy milk production, it is in polygenic nature. You are correct, but in case of the meat, it is still governed by this one gene.
Okay, sir. I have other doubt too, sir.
Yeah, please go ahead.
Sir, is there any studies ongoing for producing a heat resistant? Now most of due to climate change, most of the animals are prone to heat, susceptible to heat stress. So is there any ongoing studies in heat stress?
No, we here at the India is not yet, but I think we submitted one grant already on that direction to making the climate smart dairy animals. So we have been manipulating these target to manipulating this melanin synthesis pathway. But one study has been published, I remember, from the Well Laboratory, DN Well Lab. If you, I don't know, the DN Well in this New Zealand. So
yes,
okay. So DN Well has been done. What he done, he convert the black color into the gray color. The HF
Yeah,
cattle, if you know, the black color HF cattle patches are there, so they convert into the gray color. So that's where also my partner in Germany, they also produce Beyond Peterson. So they also produce that cattle which has a climate acclimatized and resistance to the heat stress. So they convert black cattle into the brown color or a little bit light color. So it has been study has been done in abroad, but India is not yet initiated on that direction.
thank you, sir. Thank you so much.
Many thanks, Dr. Shilokar, for enlightening us on the topic. It's a very interesting talk. Actually, I have few questions since I'm working close to that area. So, how the knockout, or the knock-in, or is a codon replacement gene edited animal will be regulated in India? Second question about, do you speculate how the gene edited animals will be in our Indian supply food chain in near future? And in third question is about, you said that you are generating myostatin knockout in buffalo, sheep, and goat with your collaboration. Do you speculate that what about the public acceptance of gene-edited knockout food-producing animal, even if it is approved by the regulatory bodies? Thank you.
Yeah, thank you. I am not getting who is talking, but anyway, thank you. I think this is a really valid questions we need to address. So first, what are the current status? If I have noted down, if I correctly noted down, if not, kindly interrupt me. First question he has been put is that what are the regulation status in the country today? So the new guideline of the genome editing in the crops as well as in animal
Excuse me, can you repeat?
simple knockout. Yeah. Hello. Can I continue?
Hello. Yes, yes. Actually, I'm Sudip. Actually, I'm Sudip, your junior, 1 year junior. I'm just in the floor.
Panda.
So how you? Yes, Panda. Yes.
Okay.
Yes.
Okay. Okay.
Yes.
Nice to hear.
Nice to hear. Actually, I'm really excited to see your data, and this really wonderful data. So how you speculate that the gene edited animals will be in our supply food chain in the near future?
Yeah. Okay. Now I will reply one by three questions you have asked. Regulation as on today, there are categories of the animals. Now department has been published in 2020-21. The category 1, category 2, and category 3. So myostatin kind of the research or simple knocking out or point mutations or the couple of nucleotide chain, they have been category 1 and it is easily regulated. So there is no need of much regulation required from the IBS, RCGM or GAC. So Institute Biosafety Committee, it can approve that is a point mutation while if your aim was to generate the point mutation which you have been done in the myostatin. Regarding the supply chain, recently, I think a couple of months back, the Department of Biotechnology, or may say Government of India, has been excluded OSD1 and OSD2. You might be aware that in case of the crops, so it has been allowed to use for the cultivation and the supply chain. So this has been OSD1 and the OSD2. So and we hope in case of the animal too, similar line will be adopted for this using of this one. And regarding the public acceptance, as on today, if we talked about the farm animals in the country, we not yet produced any kind of the animal yet. So our first target was to produce the animals, demonstrate that this is possibility, then we can approach to the government to make a guideline and to have an opinion from the public that they are acceptance or not. There is a debate in entire world is that it is an acceptable, not similar to cloning. Except the European Union, rest of the world still they are using the cloning technology for production of the animals. And the India also now recently the Indian Council of Agricultural Research recommended this technology for the breeding purpose, and hope in coming years this technology, the cloning technology, will be used for the breeding purpose, particularly clonable semen. First one, our aim was to produce the animals and demonstrate here, and then we have a public opinion on that. Is that really they would like to accept or not? But since we don't have not much animal or not a single animal has been produced for the genome edited transgenic animal, that is a reply from my side.
I have one question, if you have time, or if I may, last question.
Yeah, please go ahead.
So what is the success rate of zygotic microinjection in sheep or goat or in buffalo at your hand or with your collaborators?
So zygotic microinjection in case of the sheep and goat, it has not been tried yet. So they have been very early stage because this project has been initiated last year only, the sheep and goat collaboration. But I can give my opinion on the buffaloes. In case of buffalo, if you talk about the editing efficiency, we got around the 10 to 15% editing efficiency. So it works well. But in zygotic microinjection, we have been a bit worried about, despite having the editing efficiency, if there is a mosaic animal, then we may have difficult for us to generate the line, means homozygous line. So that's why we are more focusing on generation of the single cell lines having the biallelic editing through electroporation and the transfection in the cells. And once they have cells with us, we will use for the, and we will use for the SCNT because that is well established in our laboratory.
Thank you very much, Dr. Shilokar, for fabulous work and showing all these things. Thank you very much.
Thank you, Sudipta.
Thank you everyone for your questions. If there are any more other questions, please feel free to raise your hand, or even later on we'll have another question and answer session, so you can ask your questions then also. And now, before we move on to the next session on funding opportunities available at India Alliance for all the researchers out there, we'd like to take a short break of 5 minutes, and we'll be back with Ms. Isha Goel, who will speak as a grant advisor, will speak to you about the funding opportunities. So a 5-minute break and we'll be back soon. All are back from the break. And now we'll move into another important session of the day, which is the funding opportunities at India Alliance. So let me introduce Ms. Isha Goel to you. Isha is a nanobiotechnology engineer possessing and possessing the MTech in nanobiotechnology from IIT Roorkee and BTech in biotechnology from MIT Noida. She has ardent research experience, especially in biomedical domain, which translated into a patent for burn wound healing patch. She has also worked as project executive in biotechnology firms in Delhi, where she was part of management team for National Plant Tissue Culture Certificate System, a training program for African candidates, and setup of new ventures. She is currently a grants advisor at India Alliance, and she will be talking about the various India Alliance funding opportunities available here. Isha, go ahead with your presentation.
Thank you, everyone. Thank you very much for the warm introduction. I'm audible and my slides are visible?
Yes.
Thank you, Himashri. So I'm Isha, Grants Advisor at India Alliance, and I will be talking about the various opportunities that India Alliance provides you all to advance research in India. So India Alliance is a public charity that funds research in health and biomedical sciences in India. We are funded by Department of Biotechnology, Government of India, and Wellcome Trust UK. We invest in transformative ideas, support research ecosystems to improve animal and human health and well-being. Since the launch of India Alliance in 2008, we have successfully stirred fellowship programs, which was started in 2008, and clinical and public health fellowship programs, which were launched in 2013. In our phase 2, which started in 2019, India Alliance launched new grant programs, which were promotes clinical and public health research through clinical and public health research centers, and collaborative interdisciplinary research through team science grants. The main focus of India Alliance is to support training and building capacity in biomedical and health research, strengthening research ecosystems in India, fostering international collaborations and interdisciplinary research, and making research open and accessible for all. To date, we have funded individuals, institutions, and projects across India. This map shows our pan-India reach, where each circle size depends upon the number of institutions that are being funded in that area. Since 2009, we have given out 537 awards in 133 organizations across 47 cities in India. As I just discussed, I'll briefly take you through the features of our flagship fellowship programs first, and then move to our new grants programs. So, with India Alliance fellowship programs, our main aim is to implement globally recognized grant funding model and maintain international standards. We aim to nurture outstanding biomedical scientists and promote clinical and public health in our country. India Alliance fellowship programs, to reach these aims, we have attractive inbuilt features. Like in India Alliance fellowships, the emphasis on the lead investigator. There is no restriction based on age or nationality. We give due regard to non-research career breaks, provided they are justified properly in the application. So, what does a non-research career break mean? Like, there can be unprecedented circumstances like the pandemic or maternity reasons. So we give due regard to these. A salaried position or commitment is not a requirement for our fellowships. We use an online, secure, user-friendly application process through ISS portal. We encourage fellows to pursue interdisciplinary research, and our awardees are given provisions to develop international collaborations. Selected applications through our application process go through the international peer review, where applicants receive insights and suggestions on their proposals. The most important feature is that the quanta of funding we provide is generous, and fellows have a lot of flexibility in how they utilize their awarded funds. So, our fellowship programs provide funding for individual scientists at different stages in their careers. Early career fellowships are designed for talented, newly qualified PhDs and postdoctoral researchers to attain independence. The scheme provides a unique opportunity for them to make an early start in establishing their own research program. Intermediate fellowships are for researchers who wish to establish an independent laboratory with a high-quality research program in India. Another thing this intermediate fellowship achieves is to bring researchers back from abroad to the Indian scientific research fold by providing such generous and flexible funding, thus reversing the brain drain. Sorry about that. Moving to senior fellowships, these are for more established researchers who have demonstrated their ability to lead an independent research project and group who would like to expand their research program in India. All these 3 fellowships are available in both biomedical research for both biomedical researchers as well as clinicians and public health researchers. You may access information related to all these schemes on our official website. Coming to the specific aspects of our fellowship schemes, these are the key provisions we provide in our basic biomedical schemes. The Early Career Fellowship is a mentored fellowship, similar to a postdoc fellowship for newly qualified PhDs, and for postdoctoral researchers, it is a way to attain independence. In line with the scheme's mandate to foster independence, early career fellows are strongly encouraged to move out of their thesis environment and continue research in a different environment. Eligibility for early career fellowships starts from last year to 4 years post their PhD or equivalent degree. This competition occurs once every year. Senior and intermediate fellowships competition is open to researchers with 4 to 15 years of post-PhD or equivalent degree experience. Applicants can choose the scheme most appropriate for them based on their qualification, research experience, and career trajectory. The intermediate and senior and intermediate fellowship scheme competition runs twice a year. As I said earlier, these fellowships provide generous fundings, and the budget cap for early career fellowship is 1.7 crore for 5-year duration, while intermediate fellowship is 3.6 crores and senior 4.5 crores for the 5-year duration. Salary is included in the budget. Fellows are provided with salary support set by India Alliance for each scheme. In case fellows receive a salary from their host institution, then a top-up is provided so as to meet the salary level set for each category by India Alliance. The fellowship can also be used to purchase equipment. Though funds requests for expensive equipment are typically not allowed for ECF, early career fellowships, it is expected that early career applicants will choose appropriate laboratories where majority of necessary equipment and facilities are already available. Senior and intermediate fellows do not have any such restrictions. There is also a provision for hiring support staff. Early career fellows can only ask for 1 technical support staff, whereas intermediate can take up to 2 support staff, and senior fellows can have up to 4 support staff, including PhD and postdoc students. Work outside host institution, like mentioned earlier, is one of the coveted provision of our fellowship, which allows fellows to carry out a part of their work in a different lab outside of the host institution. This can be in India or abroad. For working abroad, fellows will be provided with an allowance of $3,000 per month based on the prevailing conversion rate. Early career fellowships can visit a lab outside their host institution for 2 years, while intermediate fellows can avail this provision for 1 year maximum. And this option is not available for senior fellows, but senior fellows can request funds for travel and subsistence for short visits to the collaborators on their application. In addition, for travel to conference or meetings or visit to collaborators' lab for short visits, we provide up to 7.5 lakhs for 5 years for early career fellows, intermediate fellows, and up to 10 lakhs for 5 years for senior fellows. in the budget. Finally, contingency funds are provided as India Alliance recognizes the need for unanticipated costs might arise during the tenure of the fellowship. Therefore, India Alliance will provide contingency funds to cover such expenses. So this is these are the main aspects of the basic biomedical schemes. Moving on to the CPH and clinical public health schemes. I want to draw your attention to the main differences between the clinical and public health and the basic biomedical scheme. Of course, the eligibility requirement is broadened to include those with MD, Masters in Public Health, PhD, or equivalent. As clinicians and public health researchers often have a very different career trajectory compared to basic biomedical scientists, our eligibility criteria also takes that into consideration and is more flexible in terms of research experience, as can be seen from the 15-year eligibility window. As clinicians and public health researchers often have very different career trajectories, so applicants can choose the most appropriate scheme based on their qualification, research experience, and track record from the early, intermediate, or senior schemes. The salary cap and budget cap is also slightly higher for the clinical and public health researchers, mainly to compensate for their clinical practice. Since many clinical and public health schemes also require field workers, the provision for support staff is also comparatively relaxed in this scheme, though strong justification is required in the application for such requests. Our other provisions remain the same as in basic biomedical schemes. Coming to the application process to become an India Alliance Fellow. All our fellowship schemes require a preliminary application, which include the applicant's CV, 750-word abstract of their proposal, and at least one recommendation letter. This is the minimum requirement. Based on the prelim application submitted, we assess the eligibility and competitiveness in the cohort, and full application is invited from the selected applicants. Please note that full applications are accepted by invitation only. The full application is a more comprehensive form with multiple sections that an applicant needs to fill out, like it requires a detailed proposal, letters from all roles on the application are required, a detailed budget needs to be submitted, etc. Then these full applications are sent out for international peer review, which is an important step in our application cycle. The review feedback that we receive from the international peer reviewers is shared with the applicants in the end, regardless of the shortlisting decision, to help them in their future proposals. Based on the review feedback, applications are shortlisted for the interview with the selection committee and by the selection committee. Then based on the assessment in the interview, applicants are chosen for award or decline. This entire cycle takes 10 months to complete. The important thing to note is that once you have submitted a full application, you cannot compete again in the same round or a scheme below a junior competition. So if I will explain it with an example, if you have applied for an intermediate fellowship, you cannot apply again for an early career fellowship or an intermediate fellowship, regardless of the decision on the application. You can, however, apply for a senior fellowship. This resubmission policy applies after the full application is submitted. There is diversity in our awards, and we have a diverse pool of awardees. We meet gender challenges in science by adopting policy changes. Like I mentioned earlier, we consider non-research career breaks, including maternity breaks, during eligibility checks for our applications. Our fellowships also have the provision for a 1-year full cost extension to fellows on maternity leave on their term. Nearly 36% of our awardees are women. We meet, if you look at our fellows, they are divided, how our fellows are divided into the two schemes. 75% of our fellows are working on basic biomedical research problems, while 25% are working on clinical and public health themes. Our fellowships are also extremely competitive with a success rate of nearly 10.6%. Proof of that is how out of 4,497 applications, 479 have been awarded. Several of our fellows apply from outside India, and nearly 60% of our awardees hold faculty positions now. Which is a proof of reversal of brain drain. Also, 7.5% work at new institutions. These are institutions which have been established after 2010, which is which shows the positive impact India Alliance Fellowship is creating on Indian research ecosystem. So keep an eye out for our call for applications on our website and social media handles. Check eligibility and submit the application through our online portal on ICES. Moving on to the second phase, which is India Alliance grant schemes. Having covered the fellowships covered by India Alliance in some details, I will now talk about the funding schemes which were started in 2019, the Clinical and Public Health Research Centers and the Team Science grants. So, introducing Team Science grants. Team Science grants funds teams that bring together scientists from multiple institutions to address an important health challenge in India, while clinical and public health research centers are envisioned as virtual research-oriented centers focusing to improve clinical and public health research themes. They aim to build clinical research capacity and develop physician scientists. These clinical and public health research centers have an embedded clinical research training program, CRT, which aims to develop physician scientists in India. Elaborating on the Team Science grants, this grant funds teams of researchers who bring together complementary skills and resources to address an important health challenge in India. These grants fund high-risk, high-reward research work. The project should be multi-institutional and interdisciplinary in its approach, which is suitable research management plan and structure. A minimum of 3 investigators should participate, including 1 principal investigator who would manage it and lead the team. The applying team should bring different expertise to address the research problem. No more than 2 investigators can be from 1 institution, and a minimum of 2 institutions should be included in the grant application. We encourage gender balance and geographical diversity in the team. The funding provided is up to 10 crores for 5 years project, where funds can be requested for provisions like staff, equipment, consumables, strengthening research management structure at the host institution, etc. Moving to clinical and public health research centers. These are institutional grants that are meant to promote clinical and public health research and develop physician scientists. They would empower clinicians and public health scientists to carry out patient and population-oriented research in addition to patient care and preventive medicine. The CRCs are envisioned to include more than one institution. Strong justification would be required for a single institution CRC application, in which case multiple departments at that institution should come together to build the application. Here also, the funding provided is up to 10 crores for the 5-year project, and funds can be requested for hiring equipment, maintaining clinical and population cohorts, training early career clinicians in research, and strengthen research management structures, etc. CRTP, Clinical Research Training Program. This is a component of the Clinical and Public Health Research Center, CRC itself. Research Training Program aims to develop physician scientists as a way forward for improving clinical and public health research ecosystem in India. This is not a standalone program and will be administered embedded with the Clinical and Public Health Research Centers program itself. The program would fund 3 to 4 year mentored research training fellowship for medical graduates, MBBS, and postgraduates, MD, MS, or equivalent. CRTP aims to integrate basic and clinical public health research, provide training in research design, research ethics, and statistical analysis through mentor training program targeting diverse areas relevant to India's health priorities. Up to 12 fellowships will be available each year, and the value of each fellowship will be around 50 lakhs, which includes only personal support for the fellow. Now that we have some idea about India Alliance grants and fellowships, I would like to very briefly talk about how to make your applications. First and foremost is motivation. The prospective applicant should be highly motivated towards his work, his research. India Alliance gives high edge to scientific, like publications, conferences attended, and for intermediate and senior fellowships, of mentoring junior researchers is desired. The question to be addressed via the proposal is a significant factor. The question must be important, relevant, and feasible within the 5-year fellowship period. Generally, researchers prefer to extend their PhD and postdoc work, but still, it is desirable that the applicant will come up with his or her own idea and should be able to convincingly present it. Especially for early career fellowships, it is desired that they have a willingness to move out of their comfort zone and not continue in their thesis environment. We look for good grantsmanship, which include language, format of writing, and plagiarism. Plagiarism is taken very seriously at India Alliance, and proposal with high plagiarism including self-plagiarism, will be rejected at the preliminary stage itself. Please take care of that. Support letters should not be one-liners. Letters of recommendation should explain the contributions of the applicant, and letter of support. For example, if it is from a collaborator, then the concerned person should provide details how their expertise and or how they can contribute to the said project. As we believe the proposals are ideas of the applicants and for best outcomes. The selection of the host institution, fellowship supervisor, mentors, and sponsors must be thought through. Therefore, justification for their selection should be clear in their application. So these are the main points that can be taken care of to put forward a strong application. Now here is a representation of the areas of biomedical, clinical, and public health scientists that are getting covered through India Alliance fellowships and grants. The broad remit of India Alliance Fellowship is to support biomedical research that is relevant to human and animal health welfare. Within the broad remit, our fellows are from a variety of fields ranging from molecular biology to infectious diseases to structural biology, neurosciences, public health, and various others. This layout is a representative of the various areas of our funding. If interested, on our website, all India Alliance Fellows profiles are available, and you can look through them to see what all work they are and problems they are working on. Now I shift gears a bit and talk about another one of our new funding scheme, the IRMI. India Alliance launched a new scheme to create a robust research environment by setting up research management systems. This is called the India Alliance Management Initiative, or IRMI. IRMI aims to strengthen the institutional ecosystem by facilitating setting up of research management systems through funding, training, and mentoring. India Alliance Research Management Initiative began in 2018 when a pilot study identified the need for research managers in India. In 2019, IRMI was formally launched, and since then it has been running with the aims of supporting training, career development, and networking opportunities for research managers and administrators, building national and international partnerships for knowledge and resource exchange, and creating a community of researchers in India. Community of research managers in India. To achieve these goals, IRMI offers funding opportunities to research managers through 3 types of competitions at 3 different stages of their careers. First, to launch their careers in developing research management offices, support is provided through a 3-year research management fellowship. Second, to augment their careers by supporting existing research management offices through the 1-year research management grant. Third, by providing networking opportunities to research managers through a 6-month research management travel grant, which they use to travel to international conferences and meetings. Apart from these 3 competitions under this scheme, we also have regular webinars with the idea of assisting capacity building and research management training. These webinars are also available on our YouTube channel. Also of note is our coming IRMI annual conference with the theme of connecting the dots and adapting to change. Registrations are currently open for the conference. Those who are interested can join the IRMI network by submitting the individual or institutional registration form from our website. As you can see, the IRMI network encompasses individuals across the length and breadth of our country. The overall impact of our funding schemes can be observed both on the level of an individual and as a society. India Alliance funded research has led to publications of more than 16,052 publications, out of which 1,381 are open access. Our fellows are working on policies related to use of tobacco, nutritional therapy, gene therapy, etc. And a few of these have impacted the way government policies are drawn up. When it comes to building a strong research ecosystem in the country, our fellows have trained nearly 9,000 PhD students or researchers. Apart from this, towards bridging the gap between science and society, we have implemented multiple public engagement initiatives, including popular science talks and health awareness programs, and reached out to more than 15,000 lay participants, trained several researchers in various aspects of science communication. With this, I come to an end of my presentation and would like to draw everyone's attention to the upcoming competitions. The Early Career Fellowship of competition will be launched in August 2022. Exact date will be announced on our website shortly. Senior and Intermediate Fellowship Scheme will be launched on Friday, 1st July 2022. The deadline for submission will be 28 July 2022. You can access the link for the application through our website. So we request for clinical and public and the other scheme competitions are yet to come by. So we request prospective applicants to visit our website for updated application calls, dates and deadlines. We frequently have webinars with in-depth description of the schemes, and also our grantees speak about their journey. These are also available on our YouTube channel. If interested, please have a look. To know more about our various funding schemes, do look up our website and follow our social media handles. You will find a lot of interesting stuff there, like our external newsletter brings latest updates and funding opportunities to all people who have subscribed. And our Science Together podcast series and Science Stories video series are available on YouTube channel, which highlight the impact of the impact our fellows are making in research in our society. Write to us at info@indiaalliance.org if you have any doubts about your eligibility or scheme provisions. We would be happy to help. With this, I would like to close my talk. Thank you for your time and attention.
Thank you, Isha, for the in-depth presentation on the funding opportunities. I believe the attendees may have got a understanding of if they can apply for and how they can apply for it. As Isha mentioned, I would like to again tell you to keep following our social media handles for any of such announcements. Currently, there are the Clinical and Public Health Research Center and the Teen Science grants open, and we are soon having the senior and intermediate fellowships, so have a look out for that. We have webinars, we have had webinars on them, and the webinars are available on the YouTube, and also we post it regularly on our social media channels. Apart from this, you can also write to us at workshops@indiaalliance.org, or even to info any of our email IDs that for any other information that you require. And also, if you would like any more workshops to be organized in collaboration with you. And in addition to that, we also have a small seed funding available for public engagement. So if you have any science communication or public engagement kind of programs, please feel free to write to us. Or so available. So I'll just thank you all for being here today for our workshop, and please wait for receiving your certificates. Please write to for another week, and because you write us, write them manually, so it will take some time. And an important again as a reminder to fill out the survey that you will receive now, and please keep joining our future webinars. We have 2 more webinars lectures as part of the Continue Biotechnology Education Series, so which will again take place in the next few months. So So please keep updated and join us for the future. So thank you very much to our speakers for the day, Dr. Naresh Selukar and Isha Goel, for being with us today and talking about the important topics. Thank you very much.